Good evening, everyone. Welcome to Biochemistry Simplified, another topic on genetics. I thank you all for the subscriptions. I thank you all for the comments, for the likes, for the shares. Let us keep on subscribing, sharing, commenting, so that we can all understand this journey of making biochemistry to be simple in understanding the concept behind in biochemistry. So today I'll do a slide on genetics, the monohybrid course. So if you talk about monohybrid course, you're talking about one trait. For example, in this case, we see the trait we're dealing with is seed shape. So seed shape is the trait. These traits are controlled by a gene. And we said the alternate form of a gene are the alleles. So shape, it's either smooth or wrinkled. That is the type of seed which we have. So obviously the smooth is always dominant over wrinkled. So as we said that the alternate forms of the genes are the alleles, and the trait which you are working on is seed shape. The capital R will go for the round. So the allele for the round with the capital R. Then the small r will represent wrinkled. It will explain much. That is the recessive allele. The dominant allele, capital R, will go for the round. The recessive allele, small r, will be for the wrinkled. Then we are generating a cross between two parents, whom one parent is round seeds, meaning the genotype is homozygous dominant, capital R, crossed by wrinkled seeds, small r, homozygous recessive. So the male and female will be able to produce the gametes. So at that point, we are talking about the capital R, assuming it's from the male, small r is from the female. So assuming the female is homozygous recessive, the male is homozygous dominant. Then we talk about parental generations. The parental genotypes will be homozygous recessive, homozygous dominant. Then in the box, in the 4 boxes which are in black, those are the first filial generations, or the F1. So we'll get the capital R from one parent and the small r from the other parent, which will result in heterozygous. Same with the second box, the third box, and the fourth box. So all the F1s, the genotypes will be capital R, small r, or heterozygous. The phenotype, they will all appear round because the allele for round is dominant over allele for wrinkled. So the round allele will suppress the wrinkled allele. So the phenotype which will show for such F1s will be they appear all round. Remember, we mentioned that behind every phenotype there is a genotype which is responsible for those physical expression. So we have to start with the genotype, and we'll have the genotype all being heterozygous for the F1s. It means that the phenotype, the physical expression of the F1s, they will all appear round. So we talk about genotypic ratio, all alike because they are all heterozygous. Phenotypic ratio also all round or all alike. So in that case, we have a monohybrid cross. We are dealing with one trait, and then we have the 2 alleles representing the dominant allele round, and then the small one, the small r, representing the recessive allele for wrinkled. So we have the parental generations, we are talking about monohybrid. So we have to recollect whatever we've just seen in the previous slide. Offsprings all heterozygous. The F1s all are heterozygous. Capital R, small r. Heterozygous, the other word of heterozygous is hybrid. For the homozygous recessive, homozygous dominant, we refer to them as pure breeds. So the heterozygous are known as hybrids. So we found that all the offsprings, the F1s, the first filial generations, they are all heterozygous. So we talk about hybrids. So these offerings are known as the F1s, or the first filial generations. So the genotypic and the phenotypic ratio is all alike. The genotypic ratio, they're all alike because they're all heterozygous. For the phenotypic ratio, they are all round. So you talk about the phenotypic and the genotypic ratio, all alike. From the F1s, we can also self them, and then we'll be able to generate the F2s. So we are still on seed shape. You have to specify that it's still a monohybrid cross. The alleles, we are still standing with the capital R for round, small r for wrinkled, but now you are crossing round seeds and round seeds from the F1s, where we had all the heterozygous. If they are self, then we have round seeds and round seeds, meaning both parents are heterozygous, as showing on the Punnett square. So you will have the parental generations. These are the F1s now. If they are selfed, then now we are going to have homozygous dominant in the first box, 2 of the heterozygotes, and then we are going to have homozygous recessive in the last box, such that we are talking about the genotypes. All the genotypes are going to be represented: the homozygous dominant, the heterozygous, and the homozygous recessive, such that for the phenotypes, we will talk about 3 of them will appear round, because the law of dominance, which we'll look at, the dominant allele for round will suppress the recessive allele for wrinkled. So in such cases, the phenotype will take the allele which is representing the dominant allele. So they will appear, the heterozygous will appear round, and then we will have 1 wrinkled. So in this case, the genotypic ratio is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. It means the phenotypic ratio is 3 to 1. The 3 are the round ones, and 1 is for the wrinkled. So you just know that the genotypes, we have only the homozygous dominant, homozygous recessive, and heterozygotes, and they're all represented here. When the F1 are selfed, the F2s, in the F2s, the second filial generations, we are going to get genotype, homozygous dominant, homozygous recessive, and heterozygous. For phenotype, we'll both have the round and wrinkled, which makes the genotypic ratio to be 1 to 2 to 1, while the phenotypic ratio will be 3 to 1. So this just shows much whatever the previous slide has talked about. The parents from the F1s, the crosses were generated between 2 of the heterozygous, and then the offsprings, it means the F2s, we already got 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. They are referred to as second filial generation. And you've seen the genotypic ratio and the phenotypic ratio go to 1 to 2 to 1, 3 to 1, respectively. A test cross can also be conducted. For test crosses is where an heterozygous or hybrids are crossed by either homozygous recessive or homozygous dominant. So it is known as test cross. So the possible test cross are homozygous dominant crossed with a hybrid, that is heterozygous, or homozygous recessive crossed with a hybrid. So in that case, for example, if you have still monohybrid cross, the trait still is seed shape, capital R for round, small r for wrinkled, the cross between round seeds and round seeds. It means the homozygous dominant are still round seeds, the heterozygous are still round seeds. So we have the test cross of the hybrid and homozygous dominant. So you'll put the parental generations there so that we can get the offsprings out of it. So you will have 2 of the homozygous dominant, 2 of heterozygous, such that the genotypes which you will have will be capital R, small r. Phenotypically, they will all appear round. So genotypic ratio is 1 to 1, meaning 1 homozygous dominant, 1 heterozygous. For phenotypic ratio, they will all look alike. They will all appear round. Remember, we said the genotypes are the ones which will control the phenotype. So behind every phenotype, there is a genotype which controls it. Still, we are talking about seed shape as a trait. Alleles are still for the capital R for round, small r for wrinkled. But now you are doing a test cross, the hybrid using the homozygous recessive. And in that case as well, you will get 2 of the capital R, small r, 2 of the heterozygotes, and 2 of the homozygous recessive. So you'll talk about the genotypes, capital R, small r, that is heterozygous, small r, homozygous recessive, brings the phenotype. You'll have both round and wrinkled. So the genotypic ratio will be 1:1, while the phenotypic ratio will also be 1:1. Remember, the homozygous recessive are expressed only when it is in homozygous recessive state. When it is in heterozygous, the homozygous recessive is not going to be expressed. Whatever is going to be expressed is the allele which is responsible for the dominance, in this case round. So in that case, the capital R, small r will show the characteristic for the dominant allele, while in the case where both of them are homozygous recessive, it will express the allele which is responsible for the recessiveness. Still, it talks much about whatever have been crossed, homozygous with a hybrid, heterozygous, you will get 50% of the homozygous dominant, 50% of heterozygotes, or 50% homozygous dominant, 50% homozygous recessive. So you talk about a ratio of 1:1. So it talks about results of the homohybrid cross. Inheritable factors or genes are responsible for the heritable characteristics. We already mentioned that the genes are the ones which control the phenotype. The genotypes control the phenotype. So the genes will always control the expression of a certain trait. So the phenotype is based on the genotype, and each of those traits is based on the 2 genes. One has to come from the mother and the other one has to come from the father. And now we are talking about true breeding individuals who are homozygous. Both alleles are present. So if you talk about true breeding, we talk about the pure breeding, homozygous dominant or homozygous